host, Nick Jikomis, and today my guest is Dr. Lee Cronin. But before that, a couple housekeeping items. If you're a regular listener, you may have noticed that the name of the podcast has changed from Good Chemistry to the Mind and Matter podcast. While this may be a minor nuisance for some of you that have been listening for a while, I had good reasons for the change, and the most important thing is that nothing else about the podcast is changing. I will continue producing discussions on a wide range of science-related topics, especially in my core areas of interest, which tend to be things like how psychoactive drugs affect the mind and the brain, the biochemistry of life, and how emerging biotechnologies interact with it, as well as questions in the general area of cognitive science, including how brains give rise to perception, behavior, and mental phenomena generally. For today's episode, I spoke to Dr. Lee Cronin. Lee is a chemist from the University of Glasgow in the UK. His scientific work focuses on the chemistry of complexity, especially as it relates to the origin of life and our ability to detect signatures of life in the universe. We discussed a variety of topics related to the question of what life is, how and where it arises, and this included discussion of assembly theory, which is a framework that Lee and other scientists have been working on to better understand what life is, how to find it elsewhere in the universe, and even how to create it in the lab. We also touched on how some of these things connect with ideas related to information and consciousness. It was one of the more interesting discussions I think I've had so far on the podcast. And as always, if you enjoy the content you hear, please like, share, and subscribe. Today's show is brought to you in part by Dosist, an all-natural cannabis company specializing in dose-controlled cannabis products made with plant-based ingredients. To learn more about Dosist, their products, and where they are available, please visit their website through the link in the episode description. Hey everyone, I want to take just a minute to tell you about an app I am partnering with called Readwise. Readwise is an app that organizes and helps you get the most out of your digital highlights. I use it to organize all the highlights I make in my digital books on my Kindle, and so if you're like me and you make a lot of highlights and you like to revisit them often to refresh your memory, Readwise is the perfect app. You can also take photos of any physical books you've highlighted and upload those. It also has cool features that allow you to share your favorite highlights and quotes from books on social media, and it syncs with note-taking apps like Evernote, Notion, and Roam. You can tag, search, and organize your notes and highlights on Readwise, and it helps you connect ideas in new ways and retain more of what you read. So if you click the link in the episode description, you can get Readwise for free for 2 months when you sign up for their annual plan. That plan is only $7.99 per month, and it's a relatively new app, so they're adding new features often, and if you sign up for the annual plan today, you can lock in that price, which will stay at $7.99 even if the price increases in the future as they add more. So if you do a lot of highlighting and note-taking and you want a good way to organize all that information, check out the link in the episode description. And with that, here's my conversation with Dr. Lee Cronin. Professor Lee Cronin, thank you for joining me.
It's great to be here.
Can you do a brief intro for people in terms of who you are and what you do at a very high level?
Yeah, I'm an academic at the University of Glasgow in a school of chemistry. Although I'm trained as a chemist, but really I would say that I'm a theoretician that can only do theory by doing experiment, which seems a bit weird. And my medium of choice through happenstance happens to be chemistry and stuff I can touch. And I'm interested in control of matter from the top down and like human beings and how life got going. So from the bottom up and how information gets processed in the universe.
Interesting. So you do a lot of theoretical work, you do a lot of chemistry work, and I want to get into some of that. But a lot of it does center on this very broad, very deep question of what is life? And I thought maybe we could start by having you maybe paint a brief picture for people of the intellectual history of this question. Who are some of the major thinkers and ideas that have grappled with this question that have perhaps influenced your own thinking?
Okay, yeah, that's a huge question. I mean, it goes back to antiquity, right? When people were trying to figure out what, the ancient Greeks were thinking, the Bible, of course. So I won't go to the Bible or the ancient Greeks because I'm just not a very good scholar of ancient history. But I would say that in terms of who have influenced me, I think that I was I've been influenced by, I suppose, Charles Darwin. If you read his book, it is really interesting the way he kind of this thing that's evolution just becomes self-evident after a while. I think some physicists have influenced me in terms of Schrodinger, Einstein. Schrodinger a bit more curious because he thought that he understood what an unsolved problem of life is. And then I suppose coming on to today, Freeman Dyson, I found really inspiring. A very interesting lady at the Weizmann who's still alive and kicking, Ada Yonath, who she got the crystal structure of the ribosome. She has influenced my thinking a lot. Sarah Walker, ASU, Arizona State University, has influenced my thinking on that life is more than just stuff. There's something else, but it's not magical. It has to do with information, or what we might call information. Yeah, there's lots of, and I suppose Newton actually, for just being basically very odd person, clearly very lonely intellectually, and just playing around with reality and himself. And I think that, Newton kind of was an alchemist who happened to do some physics. But he was a pretty nasty person if you look at the history books. So he has influenced me, not because of his nastiness, but just because of his the way he framed it, And so, yeah, but really right now, there's a lot of interesting people interested in this problem, but they are all, I would say, not seeing the elephant in the room. If maybe we don't either, or the life in the universe.
And so I want to maybe start to talk about let's talk about first what all life forms have in common on Earth. So what are the core features of life as we know it on Earth? And how does that maybe tie into how you start to think about how life arose?
Yeah, so let's start from a very basic materialistic point of view. All life on Earth, as we look at it, it has carbon in it. So that's one thing. There are common elements. So I think you could say, I have my periodic table mug here. So having carbon, nitrogen, oxygen, sulfur, and phosphorus, and some chlorine is important. But I think that if you were looking at life on Earth from a naturalist point of view, and you had a microscope and you had a video camera, you would be able to say a number of things about life. Is that some things move around and some things don't. But if you look at the things under the microscope, you can see a cellular structure. And if you're fast enough to capture the things that moved around, you'll also find they have a cellular structure. And if you looked at seawater or even river water or anything, and you looked in the microscope, you would see cells. So that you would, I suppose, just from looking through your eyes, you'd say, well, there's stuff going on. There's these things that look like capsules or packets or whatever, and they appear to be doing stuff. So I think that is kind of the starting point, isn't it? From a very phenomenological point of view, that life appears to be these things. And if you wait some time, these things move, divide, and appear they're warm, right? They give off heat. So, and if you take away the fuel, or what we might call fuel, they no longer give off heat, and they no longer move, and they no longer replicate. So there's something odd going on with this matter. This thing is able to autonomously wander around and take resources from the environment that doesn't look like itself, and then manufacture more of itself. And if you think about it like that, that's kind of weird, isn't it?
Yeah, just the basic idea that you've got matter and it's got a particular shape, essentially, and somehow it's reproducing itself. It's causing more stuff to look like it. You've talked in some talks online about this notion of selfish matter, just sort of generalizing the term, the famous phrase from biology that there's a selfish gene, that genes are these sort of units of replication, perhaps the smallest units of replication. And I'm wondering if you could talk a little bit about your idea of selfish matter and whether or not genes are the smallest unit of replication, or what the smallest unit of replication might actually be in principle.
Yeah, I think so. Selfish matter is a good one. And also, I mean, not just selfish, but some matter's lucky because it's animated, right? It gets to go places other matter doesn't go. And just to give you a segue before I answer that question, I wasn't really that fascinated with biology growing up. was fascinated with physics and chemistry and mathematics and computers. But now and then, when I stop and look at a wall and there's a piece of grass or something growing out of it, and you think, and you know what's there, right? This replicating machinery, this ribosome just growing there. For me, it's like a source of wonder that I don't think I'm ever going to shake. It is so incredibly interesting that there are ways that life is kind of characterized by these little capsules of matter that are minimally, they're islands in their own right. They can go away and just terraform their surroundings. So yeah, going to selfish matter, I suppose that if you think about what evolution is doing, and I suppose that by calling it selfish, I'm anthropomorphizing. And I suppose actually it's not selfish matter. There happens to be matter that's able in the current environmental conditions to be successful. And we don't even know that's anthropomorphizing. So what I'm meaning is to say selfish, successful, we can't do that. Okay, let's say persistent. Well, persistent is kind of, okay, just there and able to reproduce itself. And so what you do is you go from this selfish to this kind of functional object, and you can start to realize that through time, this matter is able to maintain its form, repair itself, and reproduce. And so there is some processing going on. And I think it's fascinating that where that matter is, if you almost acts like a singularity in space, where it's drawing fuel and stuff through that point. And something's happening in that point. There's some information, there was something going on where it's able to take this dead stuff and literally, make living soul of it. So actually, I shouldn't say living soul. What I mean? Give it the essence of life, right? And we didn't know what that was hundreds of years ago. We now know what that is in terms of, producing cells, and in those cells, there's DNA and RNA and stuff. So that's kind of my notion, is that you have this inanimate stuff, which has maybe some chemical wealth to it. It's got the right bonds that you need, the right elements. And then I can procure that and extract resources. And those resources I can use to build more of myself and to also kind of do something interesting. That I've got this resource. I'm going to consume this resource. I'm going to metabolize it, build with it. But then when I go through the process of replication, I'm going to allow there to be not a faithful, all I'm going to do is the best I can at making a proper copy. But if some errors come in, I'm going to tolerate that, because maybe those errors will act as a probabilistic program to make my new offspring better, fitter, more able to survive in the environment that may or may not be changing. And so you have this really interesting search algorithm that animated matter with selection is able to basically record what's going on in the environment over time. And that may be argumentative. It might be the all animate matter. If you pick one of your atoms, that's animate. So at what level does that atom become animated rather than inanimate? Well, it's in the network of surrounding objects. So that's a rather complex answer to the question, but I think it provides us a lot of points to dig in and really explore what we mean by the living state. Because if I took one of your carbon atoms and ran away with it, you wouldn't be that unhappy, because you have a lot of carbon atoms. So we know that life isn't just in the atom, it's in some other aspect. And I love to play games with chemists and biologists and physicists and say, well, if we all agree that's the same atom, where is the life?
Mm-hmm. What is the importance? It seems like one of the most important features of these self-replicating patterns that we call life is their encapsulation, that there is a cell, there's a separation of an internal environment from the external environment. Could you speak a little bit about just some of the basic biology there for people who might not know about, lipid bilayers and things? And what do you think the importance is of having that bilayer, or that cell that separates an internal from an external environment? And is that strictly necessary?
Yeah, so it kind of also amplifies what you're saying in the last question. I realized I didn't answer entirely, like the minimal unit of life. And so you suggested, is it a gene? And let me answer. So let me just carry on and then go on to the cell. So a gene is not a minimal unit of life, actually. It's not the minimum thing that's selected upon. That people, it is a unit, but as you're alluding to right now, objects, molecules are not alive. Genes are not alive. It may look like they're alive. I can, as a biotechnologist, I could evolve that gene in a laboratory. Is that the minimum unit? No, because the gene needs me to be enslaved to it, moving my syringes around, doing stuff. So you're like, oh gosh, so if the gene isn't selfish, what really is selfish? And I think that then what we've got to do is zoom out and say, what is the minimal viable unit of biology on Earth? And that's what you've just kind of said, well, let's talk about a cell. So a cell is, so what is it? Let's say the minimum viable unit in biology appears to be a cell. And what is a cell? A cell is literally an object which has an inside, which is to some degree protected from the outside world. So what does that mean? Well, that means the contents of the inside has a fairly good memory. A cell is a bit like a book, but it's a bit more than a book, because a book, a reader has to come and read it, but the cell can read it. So a cell is a book that can read itself. You see what I mean? So you have the contents of the book, and if the book could read itself and then act on it with the information, then suddenly that's what a cell can do. And the reason why the cell needs a boundary, so it could be a lipid bilayer, and a lipid bilayer is simply a bit of soap, if you like. There's a head and there's a tail, and the water-loving heads will basically gather together to make one type of leaflet, or they'll go the other way around, depending on whether you've got reverse micelle or micelle. And the greasy part will just form this lovely layer. And so it will act as a barrier between the inside and the outside. But not only does it act as a barrier, it allows some movement of stuff from the outside world to the inside world. And the inside of the cell has an elaborate control mechanism which has been evolved to basically respond to the outside world, but not too quickly, so you can get stuff in and out. So really, the cell is the most minimal viable unit of evolution. But it's not quite that, because not everything can survive, and it's all a bit on its own. Because what I mean is, if I had an individual cell, is it viable? Yes, but maybe you need to give it some amino acids. Maybe it needs some oxygen. Maybe it needs some CO2. Maybe it needs some sugar. Where does that come from? And then you start kind of getting this really weird, mind-bending, well, what is the minimal unit? And then you come to the conclusion, well, there is no minimal unit, but there are different types of object which are more autonomous than others. So a bacterial cell is very autonomous on Earth. It can go anywhere, find food. Viruses are not so autonomous. They have to get in the cell to do their thing. And I think that the cell is characterized by a number of objects. So I'm actually answering your questions because I realize I go off at tangents each time. So there is some genetic material in the cell, which is explicitly written down and what something we would call a code. People claim it's a digital code. I don't think it actually is, but okay, we can talk about what that means. There is a metabolism that goes on, which is able to basically provide, open up and close ion channels. There's literally a pump. I should view the cell a bit like one of those bouncy castles that you have to have a pump keeping blowing up so the kids are happy. And basically, for the bouncy castle to remain viable, you have a pump that's blowing air into it continuously. So a cell is a bit like a bouncy castle. It remains inflated, but as long as the machinery is keeping it inflated, and that requires glucose or some kind of sugar. So it needs to be inflated. There needs to be ions going in. There needs to be raw materials going in. And then the cell's also responding to signals from the outside. And then as a response to those signals from the outside, it may change its internal mechanism. So it may go defensive, or it may start to say, hey, there's plenty of food. I'm going to replicate now. So I'm going to make another copy of me because times are good. And why not? That's what's supposed to happen. And it will make a copy of itself. It may be that it's a liver cell. So if it was an organ in me, it got a drug to detoxify. It would do its job as a liver do, which would detoxify that, make it water soluble and do its thing. So I think that the cell really has a role in its in the function. And there's also a support system to keep that cell viable so it can repair itself as well, or copy itself. So they're kind of all the different things that can happen. But the interesting thing is the life cycle of the cell. The cell, when it copies itself, that process in populations is where you read information from the environment into the cells. The cell very slowly learns about the environment over time. And that's kind of the mechanism of evolution that we can discuss.
And how closely tied to the notion of what it is for something to be alive is the concept of Darwinian evolution? I think it seems like most modern thinkers today would pretty much say that those two things can't be separated, that if something is alive, it is capable of undergoing Darwinian evolution. Do you view it that way? And
yeah, I would say that everything alive has come through a causal chain of evolution. Or everything that maybe is living. So my friends Sarah Walker and Michael Lachman wrote a really nice piece called which is a beautiful piece, but it's still confusing because they talk about life and alive, and I would rather talk about living and evolving. So I would say to you, all right, Nick, have you got kids? Do you have to intend to have kids? Are you having, and you'd be like, what? So, well, I want to know if you're just simply living or if you are evolving. Mm-hmm. You're like, okay. So really, living things are capable of evolving, but not all living things have to evolve. There are dead ends, right? There are some people that choose not to have children. There are some species that are the product of a union of two other. I always get the donkey and the mule mixed up, but basically there are some things that are no longer fertile, and therefore, you wouldn't say you have no animal rights for that object. You wouldn't abuse that object more because it wasn't capable of evolution any longer. Mm-hmm. You would treat that with the same respect because it's a sentient being. Sentient being. It's a conscious animal, right? Mm-hmm. I have to be very careful with my terminology here.
Yeah.
So I think that. So,
and I suppose too, if you think about like the very beginning that we'll talk about, the origins of life, there could have been many dead ends that sprung up before something sort of took.
Oh yeah, absolutely. And so I think that really evolution doesn't care about what is living; it cares about what is evolving. As I say to some people when I'm playing games, like I said, look, life is difficult. We evolve together, you die alone, right? So and what I mean by that is that evolution doesn't really care about your death per se, other than your loss to the population. But what evolution does care about is how you contribute to the population in the environment selecting going forward. So obviously the act of death, it can be important because you might release resources, you might do something else. So I think there is this chain. What I'm really interested in is causal chain going back to Luca, where you are acting and the information that you've gathered, you push into the future. But you could build artifacts in your life, these podcasts even, right? That they will go on and influence other people, right? That would, in a certain way, that they wouldn't have. They may do things as a result of hearing to a podcast that he would not have done otherwise, and affect the very future of the humanity. And so, in a way, that we are now transcending. We are above that type of evolution because we're in this technological world where we can communicate in lots of different ways. So, evolution is very good for biology, but as for technology is another layer on top of that, and can have global influence in a different way. So, and technology is alive. It is living. And people don't, and this is where the integrated information people, when they're talking about consciousness, confused everybody else because everything conscious. Well, actually, consciousness is also a distributed phenomena that we can talk about later if you want. So it's really important to understand that chain of reasoning and that chain of data. What really interesting to me, at some point, we may be able to produce some informational artifacts that we can get off Earth, that we can send to Mars, and then maybe able to terraform Mars and put life on Mars that's got nothing to do with the physical, biological causal chain of life on Earth, even though if it wasn't for us, there would be no Martians. But that's going off a totally different tangent, maybe for later.
Yeah, I do think I want to circle back to this connection, I think, that maybe many people don't make, but some have, between questions like the origin of life and questions like the origin of mind or consciousness, because there is this replicative quality to mental activity that is really interesting to think about by analogy with things like genetic evolution. You mentioned a term that I want to unpack for people, though. You mentioned LUCA. And so I'm wondering if you could just describe for people what is LUCA, and why do we think there was one single origin of life for life forms on Earth?
Yeah, that's good. So I'll answer that precisely, and I'll tell you what I think. So LUCA stands for the last universal common ancestor, and it's a really beautiful notion, because if you look at all the cells on Earth, let's just say all the cells we know of, we look at them, and we were able to look inside, we would see some really important things. We all seem to share the same type of building blocks in proteins, in our functional parts in the cell, and our memory of the cell in the DNA and RNA. They seem to be quite similar. And obviously there's different sophistication. So some cells have more complex proteins than others. And what you can kind of do is put all these in a line and kind of infer a kind of tree of life, not just taxonomic, if I can say that, but also genetic. So you can go back. So now you can say, right, you can imagine going back. It's almost like in ancestry things you can do now, right? In our mitochondrial DNA, we know that our mitochondrial DNA came from our mothers, it came from their mothers and their mothers. And it's a really good way you can use to trace how human beings have moved around the world. So we go back, and there's less and less and less of us. And you can imagine back just at the origin of life, that there is this first cell that springs up that has all these bits and bobs ready. And it just is the seed that gives rise to all of biology. Now, that's a rather wonderful term, but I don't think there was one LUCA. I think that's rather. I don't think that's even required, because I think what we mean on Earth is that there's lots of environments on Earth which are kind of similar, and there's chemistry that's kind of similar, and there's lots of competition through phases where LUCA-like objects were coming, and it's almost like, like LUCA, or the LUCA idea, is the first expression of global democracy. And that basically everyone's like, I've got this really important, I've got this better RNA than you, but I can die occasionally. And so that doesn't work, right? So there's a catastrophic death. Or I've got this better, I'm going to use 25 amino acids, but I can't encode it properly because the error correction goes. And so there's kind of like, there was all these pre-LUCA technologies fighting it out. And in the end, you all solve the problem in the same way. It's like saying there's a LUCA was like saying there was only one point where a human being invented fire. Clearly not. It was just the conditions were right. So I think LUCA is more of an expression in geological time and chemistry on Earth, where basically the conditions were right for the minimal viable cell to emerge. And it might have happened over tens, if not hundreds of years, because you got to the point, say, a little bit of while where suddenly you had, I don't know if you remember video, you look young to me, had Betamax and VHS fighting it out, and which one would survive. And then an arbitrary decision was made, and suddenly everyone has adopted the paradigm. So I think that LUCA is merely a term that should be given as an umbrella. And I wouldn't be surprised if we find some cells on Earth, deep in the Earth's crust, that are slightly splitters just before LUCA. And they were like, no, this is our form of biological democracy. We're only going to have this signature. And because they were in a very special niche, isolated from the rest of the Earth, they could survive. And it'd be really interesting for us to find those. We are searching, but as of yet, we haven't really found many concrete examples, if any. We've kind of found some little bits. So I think LUCA is really a state of pre-biological democracy. I just made that up. Sounds quite good. I'll take a note of that for later.
So let me reiterate that to see if I'm following. So you're almost saying, basically the idea of LUCA is essentially, if you just look under the hood and you look at every cell, whether it's an animal cell or a plant cell, there's differences, but they're all basically doing the same basic stuff. We're all using DNA with the same bases. We all have the same core cellular machinery, and therefore we must be descended from a thing that had those core components. One universal.
I was going to say also, though, it is correct to say that using a process of bioinformatics and probability, you can trace for sure in some species the sequence space going back. So you'll know for sure that this species of animal gave rise to this one because the combinatorial space of genetic code is just too large. I wanted to just add that in there so to be entirely correct.
Got it. And then you're basically also saying that there was certainly at least the potential or the plausibility for a kind of convergent emergence of life. So just
Yeah.
because we infer this thing called LUCA, because the biochemistry is very similar across different life forms present today, it may have simply been that, to use your fire analogy, people, and this happens a lot in evolution, right? Similar constraints in different locations will allow the independent origin of traits. And so maybe life did originate multiple times, but it merely looks like it came from a single origin because the same set of constraints were applied at different time points at different places of the earth.
Yeah, exactly. And I think that not only that, what we might be able to see, if we were able to really play the tape all the way back, we might find that in our own cells, we have 3 or 4 different origin of life
Signatures.
common. So our mitochondria probably came from one origin of life. Our other organelles came from other origins of life, and they just got mismatched together. It's a bit like, when people are copying technologies. Oh, that's a good idea. I'll steal that here. I'll horizontally transfer it here and here. And probably the space of viruses that exists on Earth may help us replay that. But the information is so vast. We're going to have to do a lot of computing, a lot of bioinformatics, a lot of sequencing to even get a hint of that. But my guess is that we are not just the sum of 1 or 2 origins of life, but many tens, if not hundreds or thousands.
And so I think related to that notion is the question of how easy it is for life to evolve. Is it, you seem to be implying by saying that there could be many origins, so that in some sense, it's perhaps more likely than we once thought for life to actually get started. How do you think about the question of how easy it is for inorganic matter to actually become living?
So I think it's ridiculously easy, and we're just looking at it entirely wrongly. I think that the process that we're looking for, I call it, is selection. So there's a selection in the universe before biology. Biology speeds it up. And I think selection is as prevalent as gravity, right? Where there's mass, there's gravity. Where there's mass, there's selection. Okay, now you need to think about that in other terms. Say, well, okay, there's no selection going on the moon. Well, maybe there was, but the selection isn't very productive. So selection needs some processing to go on. But I think that we have missed it, and we've been so obsessive about the complexity of life on Earth. We haven't understood the mechanism which gives rise to the information storage of happenstance from the environment that is really what life is. Life is the ability to store what happened in the past, play it again, and use that to your advantage to basically animate yourself, if I was to put it rather crudely. So I think that the process of selection, everyone says, is vastly hard. One of my big gambles in my academic career, and I love taking them, my gamble is the selection. The process that gives rise to the origin of life is as common as the process that gives rise to planets and black holes and stars. So it must be staring us in the face, and I must find it in the lab.
So would you say that, in your view, life is in inevitability, that it's sort of just a natural extension of the same process of evolution going all the way back to the Big Bang, that it's just simply going to crystallize out of matter if you let the universe cool down for long enough, basically?
Yeah. That life is merely, I would say, making fun of the physicists today. The physicists today think that physics is this very small field or very big field, but very fundamental, forces, behavior, phenomena, and life's like, no, I can break the laws of physics, or I make new laws of physics that are consistent with the old ones. So it's about, I would say, life is like the blockchain of physics. The blockchain is like is all out there. You can go back to the beginning and understand it and look at all the processes, the proof, putting stuff in there. And what life does is it forks that chain of possibilities and just plays with matter. And I think that that's something that's hiding in plain sight, and we just haven't been able to see it. So there will be a revolution, I predict, in my lifetime. I'm trying to start it. I think I'm onto something, along with a few other colleagues. There's probably only about 20 of them in the world who see this way. And that we see that selection is a very, very simple thing that happens everywhere. Selection naturally gives rise to evolution. and evolution to be efficient, it just makes cells. On Earth, the most efficient way to make life is to make cells. Now, it might be on other planets where there's higher pressures and densities, there are other ways to do selection, and life looks a little bit different. One of the things that worries me is that we're so off on the causal chain. We're so forked, if you like, from the main, let's use the Bitcoin analogy, the blockchain of matter or physics, that we wouldn't actually recognize other forks. So what we've got to try and do is think more open-mindedly about what the processes are, and then go, oh, maybe that's an alien. We just haven't recognized it before because we departed from that mode of matter understanding such a long time ago. But to answer your question, I think, and there are hints in my own lab, that selection is easy to see, but we're really at the limits of analytical chemistry right now because the way it scales, I'm doing it. And also I'm fighting a huge amount of bias, not because, and this is not bad bias. This is not bad. There's bad bias everywhere. What I mean is that as a scientist, you're taught by your teachers and you're taught by the literature. So you can call that information or you can call it bias. Okay. Now, when that bias is useful, it's information, right? When that bias is kind of not necessarily proven, it's more a narrative, then it stops you from doing more work. And there is a huge narrative in chemistry about origin of life, which has stopped the field from doing anything for more than 50 years. That's why I do not work in origin of life. I work in artificial life. The origin of life is a very good discipline, but it is rife with narrative and ego. Like all science, we all have egos. I mean, I have an ego too, but my curiosity is a little bit bigger than my ego, just a little bit. But that's not maybe the case for all areas of science.
Yeah, you hinted at something that I think we'll return to, which is, in order to remove certain biases in terms of how you would identify signatures of alien life, say, one would want a technique that is completely agnostic about the mechanics of that life. You want to sort of just see some afterglow or after effect of life, no matter what it actually looks like. Before we get to that stuff, I wanted you to riff a little bit more on what are some of the major hypotheses that people take seriously today about the kinds of environments on Earth, either now or in the past, where life might most plausibly have arisen. So things like deep sea vents and things like this. Is there a sort of a short list of candidate terrestrial environments for where we think life may have arisen?
Sure. There's a short list as long as the kind of the individual cults, if you like, in the field, right? But let's try and be as agnostic as possible. So Darwin's warm pond seems quite good. Under the ocean at deep sea vents seems quite good. People are fascinated by these volcanic growths under where you've got these black smokers, these chimneys where you've got rocks and porous materials and high temperature and all sorts of stuff happening. You could imagine life occurring in aerosols in the air. I mean, it's harder to believe, but it could even occur in ice. There are some people who've seen that you can have interesting chemistry going on in ice. So you have these environmental constraints. So in the sea, on the Earth, in a cave, there's lots of these conjectures, but people say, oh, no, there's no possible on the Earth because it had to happen in the sea because all the Earth was, covered in water or whatever. And then you have all these other things going on. So it's very difficult. You can pick your hypothesis, but what then happens away from these environments then breaks into various chemical hypotheses where people say the RNA world, which is the hypothesis that the chemistry produced the building blocks of RNA, and these could self-assemble in some way. And because RNA can act as a functional molecule, can do stuff as well as store information, you got two purposes for the price of one, and RNA was did something special. Some people think that lipids came first, and that somehow you had this lipid world. Some people think that proteins came first, and some people think that chiral molecules came first. So chiral molecules are molecules that have they're left-handed or right-handed, and a chiral molecule is we've got two identical molecules and you can't superimpose them; they're enantiomers. So it's a bit. So there is a whole bunch of them, and actually it's quite funny because people like to argue about which one is the most prebiotically plausible. And I find that very amusing because of the prebiotically plausible is like, well, it's like a lawyer, right? Trying to go to the Supreme Court and say, well, I would demand precedent here. What do you think? And then it keeps changing. So there's lots. So I would say there's 3 or 4 environmental hypotheses. Okay, on ice grains as well, by the way, in crystals, in rocks. There's 3 or 4 chemical hypotheses, and then there's a mixture of all of those together.
So there's really a lot of scenarios, it sounds like, and it could be any one or all of the above. What about a slightly different question, which is if you sort of give us maybe a very abridged chemistry 101 education, if we're thinking about the periodic table, how much constraint is there on the kinds of chemistry that could even conceivably result in life? So obviously we've got carbon-based life here on Earth. People often speculate about silicon-based life because silicon is similar to carbon in certain ways. I imagine there's actually a relatively small space of elemental possibilities in terms of which atoms could actually serve as that kind of backbone of life. How should we think about this and how it relates to the question of how alien life would actually be and how it might have similar chemistry?
I think if we can go all the way back, you need, so, I mean, as long as you can describe a configuration in matter, you might have the possibility for evolution. I'm not saying you're going to get living neutron stars. That doesn't seem very plausible. We can't store enough bits. So I guess what I'm pulling back a bit because I just don't know. I'm a really poor chemist. I only know about the chemistry of life on Earth. Why? Where's my bias come from? My bias has come from one atmosphere, one atmosphere, 25 degrees C, okay? 298 Kelvin. And then the composition of material I find around me on Earth. And so all I would merely say at the moment is that biology on Earth is merely a reflection of the chemistry that was available at the origin of life and also could provide us with the right evolutionary dynamic. So, and this is about available matter. Now, you look up in our solar system and say, well, Mars looks like, a red planet. It's maybe lots of iron, lots of inorganic stuff. Doesn't look very alive. Ergo, iron is not very good for life. We just don't know enough about the environmental conditions and how fragile life is in that regard about radically changing environments. It could be that life is a phenomena that is coupled to an planetary environment. And as long as that environment is stable for a long enough period of time, you get a homeostasis on the planetary scale. And then as soon as the planet flips out for some reason, so Mars lost its atmosphere because the sun just stripped it away. It had no magnetosphere. There's no plate tectonics or anything like that. That basically life just naturally died. But that's kind of one take. If you look at the periodic table, go at your planet of choice, if you've got elements that are abundant, that are capable of covalent bonds, and you've got reasonable temperature and reasonable pressure, then carbon is good because it is able to form molecules that can store information easily. But I can imagine all sorts of other things under pressure. I mean, gosh, what could a chemist do under 200 atmospheres and a lot of temperature? They could make all sorts of phosphorus, oxygen, phosphorus, boron, what we call main group compounds. You could make interesting compounds with metals and hydrogen. We just don't know. And Titan, we're going to go to Titan in a few years. That's interesting because it's cold. There's lots of liquid alkane there. And gosh, what could happen in liquid alkane? Well, you might use a different type of very weak bonding, not strong carbon-carbon bonds, but bonds that we would call supramolecular interactions. So you might just have these kind of very weak bonds that could maybe allow an evolutionary dynamic to occur. So it really, I would say 2 things about the limitation on life. You need elements and you need configurations, and you need, where there's bonding, there's hope. So what I mean when I say life, when there are bonds, there's hope. I don't care how strong the bonds are. If the bonds are really weak, but the planet's really cold, knock yourself out. You can still have life. If the planet's really hot, but the bonds are really strong, knock yourself out. You can still have life. We should stop obsessing about our Goldilocks zone, because our Goldilocks zone is anthropomorphized just by us.
So it sounds like you would actually say that, traditionally when we think about the search for life elsewhere, people are looking for exoplanets, Earth-like planets that have the environment that's like ours. But it sounds like you're saying that we should even be much more open-minded than that, that in a
Yeah.
completely different environment, chemically speaking, you could have chemistries that give rise to life potentially that we would think of as terrible for giving rise to life in the context on Earth, but different temperatures, different pressures could allow for the use of other atoms to do things that are almost unthinkable if we're not if we're not thinking outside the box or outside the
Yeah.
planet, literally.
Outside PV equals NRT. We just got to step out of that. I think it's fascinating. I mean, if you think about it, the fascinating thing is you look at there are only four types of exoplanet. There are dead ones, and that is they're ones that maybe will always be dead. But let's not say all exoplanets. Let's say all exoplanets have the right to life one day, but let's just don't know. There's dead ones, there's living ones, there's ones that are technological, and there are ones that were once living but now dead. If you think about it, there's only four types of exoplanet out there. So when you look at these exoplanets, they will fit into one of those four categories. So we should start thinking about them being in those four categories and try and statistically map them with that in mind. Let's stop worrying about whether we exist or not. Let's pretend that out of all the exoplanets we see, they're in one of those categories. And then, it doesn't mean that there are other categories. I suppose you could have like pre-life, but you know what I mean. Broadly speaking, dead, alive, technological, always dead, or dead so far. I think that's a good set.
In terms of the origin of life on Earth, the story that you typically hear in like middle school or high school biology in the US is the Urey-Miller experiment, where essentially you just take a soup of stuff, you zap it with electricity or whatever, and you look to see, do you get living things swimming around in there, more or less? Can you describe that for people who don't know it, and maybe start to talk about today experiments that you're doing or other people are doing where, are people doing modern day Urey-Miller experiments to try and create life?
We are in my lab. I keep trying to, forgive the pun, kill the experiment, but it keeps living. I'll tell you why in a moment. So the Miller-Urey is a really simple experiment. Say, hey, we're going to take a reaction. Take some gas and some water. So we'll take, let me see if I can remember, ammonia. So that's NH3, so a source of nitrogen, hydrogen, H2, H2O, and methane, CH4. Okay, and we'll put them all together. We'll have a couple of tungsten electrodes, and the tungsten electrodes actually, which I think is quite interesting. And we'll add 50 kV to those electrodes, and this spark will go across and be refluxing. Refluxing means to boil. So all this stuff boiling up and condensing around past electrodes, and there's a cycle. So it's a natural kind of, the stuff boils, goes up. As it falls down, it falls through the electrodes, gets zapped on its way down, and you make, basically you just give it enough energy to make any old crap. If you forgive I don't know if you want swearing on the podcast, but there's not too much more other than that. So you make this stuff, and after some time, the solution changes color. Miller-Urey did this, and after a few days, they were surprised when maybe a few weeks actually, they took it out and they analyzed and they found evidence of amino acids. But in a way, I was like, it's kind of like, yeah, of course. If you think about it, if you think about glycine has nitrogen, oxygen, carbon, and hydrogen in it. You've just taken a source of these and you've blasted them together. It's a very simple molecule. It's easy to imagine. And yes, they made everything else as well. Really simple. It's like a combinatorial kind of card game, but at the time, it was a seminal experiment. It got lots of people excited. But what it didn't do is it didn't produce any complexity. It just cracked open the gases and made some organic molecules combinatorially. So it's just a bit like, literally, if you're one of these people who likes to make, I don't know, cars or yachts out of matchsticks, or you've got your matchsticks and put them together and they clumped into like 2s and 3s, maybe 4 or 5. No, there's no car, no yacht, nothing, just boring matchsticks clumped together. So then people worked on it a bit more sophisticated to try and get more stuff to happen. And they didn't really see much more. There was even a paper where everyone was so desperate to see more, they reanalyzed the old samples 50 years on with modern technology. And you know what they found? Wait for it, you're sitting down, they found more amino acids. I mean, I was like, no shit, Sherlock. Of course you could find more amino acids. I mean, it's a combinatorial explosion. Do you not understand? But it's a question of lack of ideas. So what was missing in the Miller-Urey was selection. It was just a glass bulb, right? So what I've been trying to do in my lab is to say, hey guys, let's now put some inorganic Earth's crust in it. Well, the first one we did actually is Miller and Urey. Urey actually got the Nobel Prize, but he got the Nobel Prize for the discovery of deuterium, which is an isotope of hydrogen. And so I thought it'd be hilarious just to make fun of the community to do the Miller-Urey experiment, but deuterate it. Say, what happens on planet Deuteron? And my group were like, really? I was like, come on, let's just buy some D2O. That's heavy water and do it. But when we did it, we got different results. What we showed is when the water was heavier, you basically, they move around in a different way, different speeds, and you get different compounds, which was mind-blowing, obvious to me afterwards. But before, I was like, oh, okay, yeah. You change the weight of the stuff, you have different isotopes, you get different compounds. So that was quite humble and quite great. So we did that. And what we're doing now is we're trying to put different amounts of the Earth's crust in and say, if we have Earth's crust and we can vary it, can we start to go from the soup that's unsculpted, just a mess, and can the minerals select, the crystals select some of the soup and trap that somehow and select it over time? And we're seeing evidence of that. I think we've seen the first evidence of environmental selection in a Miller-Urey type experiment. We're moving away from it because we're doing more sophisticated ones, but that's kind of where we are. There are other people doing Miller-Ureys as well, similar lines, but maybe they have a different angle where they may be changing some of the chemistry or some of the pressure or something like that.
So I know that this isn't a precise question, but is it your intuition that, in terms of the ability to create life, literally someone in a lab somewhere creating life in a beaker, is that something that you view as relatively plausible in the sense that it could very well happen within, say, our lifetimes? Or how do you sort of think about the probability there? Do you go into the lab every morning wondering, is today the day where I walk in and there's something swimming around in there?
I want to, but it's a big sociological problem with PhD students trying to get them to do these experiments, because it's kind of, I thought it was going to be easy to make a simple life form in the lab. And it hasn't been that easy, not because it's that hard, but because doing long-term experiments is difficult to set up. We have to set up all the technology. We're building almost like a large hadron collider for origin of life right now. And so now, if you say that, oh no, your life form must have a ribosome, how long will that take? I'll say, well, probably as long as it took for life on Earth, maybe a few 100,000, few million years, because the ribosome is complicated. There's only so much time. The information, you can almost calculate how much time it would take on a causal chain, which I'm doing right now, actually, which is one of the reasons why I was inspired by Ada Yonath. And that I think thanks to her discovery, and we can count the subunits, I think I know how long it takes or how many selection steps you have to go through to make a ribosome in an inorganic world, which is mind-blowing. Because then you can work out how much mass, how much time, which is like, gosh. But let's go back to your other point. The quick answer is yes, I expect to make life in my lifetime pretty quickly, but I don't think anyone will believe me because it's going to be too simple and everyone's going to be arguing about, is that life or not? So what I'm trying to do is break the problem down, and what I should do is try and break everyone's expectation down and say, well, look, let's not call it life. Let's call it evidence of selection and increase the sophistication and start to see a place where we can get selection. And then if the system starts to create a crude genetic system where you can store information and read it out functionally from nothing, then I think that I will start to convince people that this is the route to biology. But you can see how that's less satisfying than, putting a load of stuff in a bell jar, Frankenstein moment, turn it on and come back the next day and some blob going, yo, hi, I'm a Croninite, whatever.
So thinking about, you mentioned briefly something that on the one hand is pretty intuitive, I think, but I know that there's more to it that you've been working on, this notion of chemical complexity. Right. So I think on the one hand, anyone can understand that if a molecule is smaller and has fewer kinds of atoms than another molecule, it's obviously simpler. You can layer on the idea of the 3D configuration that the molecule has. But can you talk a little bit about the notion of chemical complexity and how that's relating to some of your ideas about how we detect signatures of life, irrespective of the chemistry that life actually has?
Yeah. So in trying to going back to the definition of life, I think that everyone was getting their kind of literally getting themselves overly confused by arguing about what is life. Everyone would have a new definition, right? And everyone would have a smarter definition, their definition of life. And I really found the whole thing very amusing and a little bit infuriating. And I just said, well, let's not argue about what, because people say, is a flame alive? Is a virus alive? Is a politician alive? Oh no, that's intelligent or conscious. Take your pick. And then you get your round the circle where we're arguing about what life may or may not be. And I was like thinking, well, look, what is it that life tends to do and require that makes it different to background processes in physics and chemistry? And then you're looking at the sophistication of the molecules and how big and complex they are. And the problem with the word complexity is the mathematical physicists and the computer scientists have been making complexity calculators for many, many tens of years. And they're kind of not really, they're really cool abstractly, but they don't really tell us anything about reality because their complexity calculators are complex and not computable. So I found it really infuriating because I went to the mathematicians. I was a chemist willing to cross over into a new field and say, hey, I realize there's a problem in origin of life. We're not counting complexity properly. Can you help? And they all went, nah, it's boring, or we don't care. Your way of doing it, just use this method. Just use my method. So I went away and I thought, right, I'm a chemist. I know about chemistry. What is it that's interesting? And what I noticed is that if you were to do random Miller-Urey type experiments, you get a whole explosion of stuff. Easy, because it's just the atoms will bond into each other. And I say, okay, how far do I have to go before the molecules become so big but complex, not big but not big and just a tar or a mess, but and I will say that molecule is not randomly produced. Is it even worthwhile as a thought experiment? And I played with this for a good couple of years, and everyone said, no, of course you can get complex random molecules. It just happens by chance. I said, okay, but what about a Tesla? Or back then it's probably a jumbo jet. What about a jumbo jet? Is a jumbo jet just a random? No, of course not. A jumbo jet was made by Boeing as a jumbo jet. So, okay. All right, then what about protein? They went, well, of course not. A protein is a very complicated molecule made by evolution. I said, okay. So you do agree that there are some molecules that can form that are. And then people start saying, well, no, even some proteins could be formed by chance. We just wouldn't know it's a protein, and we would call them proteinoids. And so I kept pushing, and I realized that there was this blind spot in mathematics and computing science and chemistry and combinatorics that because I know enough computer science, chemistry, mathematics, and probability theory, I was able to work out that there are indeed molecules that are so complex they can't form on Earth without the help of life. So I flipped it around another way and said, okay, this ribosome, it's not strictly a molecule. Ribosome is a really complicated thing. You would say a molecular ribosome, people will tell you off. This CO2 molecule, which one is most complicated? They want to say, yeah, ribosome. Great. So, okay, let's go down a bit. This molecule of glucose, is it more complicated than CO2? Sure. But could it rise on its own? Probably. Glucose probably could. So then I said, okay, where's the sliding rule between your CO2 and your ribosome? And I think then you just pick your point where, okay, if I found this molecule. So I kept pushing. And that's where I started to kind of realize that there was a new theory of complexity. In fact, the first ever theory of complexity in any discipline that is actually computable and measurable. And I was like, because lots of people say, oh, no, we can measure complexity, but they can't really, because it has to go through an observer. It's not objective. They have to make some assumptions. So and it's quite technical for here, but we've been playing with that, and we realized we came up with a measure of molecular complexity. Now, why does that count? Well, my hypothesis for life is don't worry about what defining life. Life as a process can create complex artifacts in abundance with multiple copies that no other process can do. And that's my working thesis. So it's a bit like I don't know, a Rembrandt or a Van Gogh. You would know the Van Goghs, Van Gogh, whatever. You would say basically the style. So when you found 10 paintings from that artist, you know it's from that artist because you recognize the configuration. So that's kind of quite an elaborate answer to your question, but it's super important because it starts to remove the bias because you don't need to know anything about the chemistry. But we could dig deeper into that in a moment.
Mm-hmm. So I do want to try and explain this on a very basic level. And this is related to what you're calling assembly theory, this idea that you can quantify quite precisely whether or not life is present based on the bouquet of molecules that you find somewhere in some environment, and in particular the distribution of complexities that you measure in that soup of molecules. And when you say complexity and how you quantify it, it has something to do with the number of steps that it would take to construct any given molecule from the elemental Lego building blocks that you have at your disposal. Is that accurate? How would you think about it?
So what we try to do is to define assembly theory. What assembly theory says, you take a given object, you say take two objects, A and B. If you wanted to basically work out which one was more complex, you would the one that takes more steps to make it, and the object is made out of bonds. So you could basically break up let's imagine, take the molecule and break all the bonds. And then you say, right, I'm allowed to form all these bonds again. What is the minimum number of steps if I'm being super lazy to do it? Now, you go, but what? It would just be equal to the number of bonds in the molecule. But come on, let's take a shortcut. When you've made one type of bond or one motif once, you've already got that in your memory. It's like, okay. So suddenly, I've made this bond once, and if I need it again, I can just pull it out of my bucket. So if I put 2 bonds or 3 bonds together and I need that exact shape again, I can do it, but without incurring a penalty other than 1 step, not the 3 steps. So that allows you to compress the information into a causal chain of events. It's a bit like if you imagine making a Lego, I don't know, Millennium Falcon or whatever Star Wars thing. I like Star Wars at the moment, my son does. There are certain boring bits where you just have to add the bricks together and some intricate bits. But when you add the bricks together, that could be in this part or another part, whatever. So you've got that part already at your disposal. So it's a bit like those breaking down. So assembly theory is a probabilistic theory to say, if I took a hammer to my Lego, broke it all apart, it would tell me the worst case scenario, the best case scenario, the minimum number of steps I would need to take to get to that object probabilistically. And that seemed really nice because then allowed us to then assess a molecule based upon the number of steps to get there.
So literally, if we were to put this in terms of Legos for people, literally all this is if you had a tub of Legos, literally Legos, you've got blue ones, you've got red ones, all of the little individual Lego blocks, and you saw a pirate ship built out of Legos, you could say, okay, there's 1,000 different ways to put these blocks together to make that pirate ship, but it takes at least X number of steps, 500 steps, say, to put things together to make the pirate ship. But for a simple, rectangle, it only takes 3. Therefore, the pirate ship's more complex.
Exactly. So if you were able to see, so the pirate ship, I would say, is a clear evidence of life. And then you say, but hang on, there's only one pirate ship. So let's say we look at Mars and you see a pirate ship, and you go, okay, it could be a one-off. But now the beautiful thing about life and living systems, it doesn't just make one pirate ship. There's zillions of identical pirate ships. So when you say, that's one pirate ship and I can break it down, that's not life. And you say, but hang on, look at my 10 billion pirate ships, they're identical. And you're like, whoa. So there has to be a process that's producing this pirate ship. And each of these steps is that information has to be encoded somewhere, because why form the pirate ship? Why not form a racing car, a Lego Santa, or whatever? I must say that when I explained this to chemists, the chemist still said, no, the pirate ship is somehow magic. And it's really interesting how in a discipline, when I was trying to get people socialized to this, they just would not accept the probability theory. They said there's something special. But I'm like, okay, if I write a book, at what point do you determine the book is written by me and not just a random type keys on typewriter. So that's different. I'm like, well, okay, and it's very interesting to kind of calibrate people into probability space. It's okay if you're a mathematician or a statistician, but chemists somehow believe in magical pirate ships.
So when you are calculating these numbers, I want to paint a short picture of how you guys do this for people who are not chemists, and then talk about like how you would actually apply this to search for and find alien life on another planet. So you guys use mass spectrometry, and you've got a way to calculate this number for any given molecule. Can you just briefly explain for a non-chemist what is mass spec and basically how does this machine work?
Yeah, so if you want, I can take a step back and say how do you do it for molecules and how mass spec works. And I think it then fits more naturally. So the pirate ship, we're talking about the Lego bricks coming together. In chemistry, we're talking about bonds holding the atoms together. So when you have a bond in a molecule, that bond basically is a specific interaction that when you look at some colors and you look at heat, when you look at infrared, that's evidence of these bonds moving. Now, if you were to take a photograph of some molecules using infrared radiation, you've got lots of different colors coming out. And roughly speaking, each color corresponds to every different bond. So a different bond has a different energy. That means it vibrates at a different place. So the vibrations of the molecule give you the colors. So if you have lots of colors associated with one molecule, it's more complicated. Count the number of colors, you get exactly the assembly number. Now let's go to mass spec. What does mass spec do? Well, in the particular version of mass spec that I use, mass spec is a way of weighing a molecule. So you put the molecule in the gas phase, and you basically have a big electromagnetic field, and the molecule moves around a curve, and you basically can calculate how massive it is. But there's more to it. What you can then do is you can then hit the molecule with some energy, and the molecule falls apart. And so the number of parts it falls into give you the number of fragments. So if you take a molecule that's really simple, excuse me, it could break into 2 equal parts. And you'd only see one part in your mass spec, so it measures the difference. So the more different parts you see, the more complex it is. Now, in our mass spec experiment, you don't image one molecule. It's not possible. You have to image many millions at once. You basically select them all so they get partitioned. So you get through your one pirate ship versus a million pirate ships instantly. In the mass spec experiment, what you do is you could always see a minimum of 1,000 pirate ships, 1,000 molecules. So you basically do the mass spec, you see the molecular weights, you see the peak of the molecule, so it's heavy. So let's take a heavy molecule. Then you hit that heavy molecule, and you know that you only selected that one molecule. It's a special surgical instrument. And then you count the fragments. And the number of fragments is roughly equal to the assembly number. And so it sounds really complicated, but it's simply mass spec, sample in, see a molecule, hit the molecule. Does the molecule have more than 15 fragments? If it's more than 15 fragments, looks like it could be from life. So that's really the kind of detector, the living detector system that we built.
So it is really, conceptually, it's as simple as if you took your Lego pirate ship and then you just took a small block of just red Legos that were just red Legos in a rectangle, and you threw them against the wall, the pirate ship would break into pieces, and there'd be more different types of pieces that you'd see because it's more complex. It's got more components.
Yeah, exactly that. And you just detect that. So that's literally it. I mean, you could also, the nice thing about assembly theory is you can use it to see if people are cheating. So if you've got someone who's playing cards and you're able to count the number of cards, you could actually see when these motifs come together, or when people are writing text, you can see because the pattern in the text has a minimum number of steps to make the words, you can use it to see if it's got a common origin, because the more copies, the more conspicuous it is. So I'm actually using assembly theory to look for plagiarism, because what does life do very well? Plagiarizes itself, makes copies of itself. And that's all it does. It tells you about those 3 variables, the information in there. But yeah, your analogy with breaking up the Lego is perfect. It's spot on. It's exactly how it works.
And so it sounds like it's obviously much more complicated than it's going to sound. But you could take a machine like this, put it on a Mars rover, put it on a spaceship, send it somewhere, and you could do this type of experiment, and using assembly theory, you could detect whether or not life was present indirectly without having to make any assumptions on what exactly that life looks like. You would simply be looking for the right distribution of highly complex molecules that you see over and over again.
Yep. There are mass specs on Mars right now. Sadly, they don't have quite the resolution and quite the ability to hit a single molecule. But we're sending a mass spec to Titan soon on Dragonfly, that what it should be. And I think it's very easy to imagine an experiment. So NASA have already sent rovers and ships with mass specs. It's going to be very easy to do it again and modify them and look for life agnostically. And what I'm saying is that if we are able to find, so the nice thing about the experiment, is the way that the mass spec, you should view it like a nose. So the mass spec basically draws molecules in to the lungs, if you like, breathes them in from the nose, and the lungs are where the detection occurs. And you really just have to get sample in the air from space into the mass spec and just work out what you've got. And so what you can do is you can then look at the complexity by sniffing the number of fragments, and just then follow the increasing of complexity to go and look for more interesting, the source of molecules. And NASA are going to do this now. I think it took me a while to convince them of this, because at the beginning, when I developed the technique, they said it was like impossible, and then it was ludicrous, and now it's obvious. So I think they're the three stages of a new idea: impossible, ludicrous, and obvious. If you see what I mean. And that's really exciting, because I think we will know for sure, because we have the scale. We will be able to send the machine to Mars, to Venus, to Enceladus, to Titan, to Io, wherever we want to go. And we will be able to detect molecules. And if their complexity is high, the higher the complexity or the assembly number, and notice I use the word assembly and not complexity, because it's comparing them within classes, but it's just like, what molecule was the most assembled? How much information did it need to get there? And the more information, the more unlikely it is that it came from an abiotic source. And yet, if you're a real skeptic, you can say, I need an assembly number of 1,000. If you're really going by what we have on Earth, an assembly number of 15. If you don't want to be in the middle, you might say 30. And what we're doing right now is checking that, doing the measurements in the lab. But the nice thing about it is you do not need to know anything about the chemistry on the planet. You don't need to know anything about the history. It's all self-consistent. There's no assumptions, no labels. Your only assumption is that there are molecules present, and that life can make complex molecules.
Mm-hmm. And one of the things I wanted to ask, and I'll still ask it, even though it sounds like you've sort of switched my thinking on this. I was going to ask how many Earth-like planets are out there, because I know that over the years they've become more and more common. We've discovered more and more. But it sounds like you're saying we shouldn't be biased to thinking about life must be in a planet that's like Earth. We can actually potentially find it in a very different type of planet. Nonetheless, can you give people a sense for how many reasonably Earth-like planets are out there and how common we think this is today?
Oh, wow. Off the top of my head, the numbers are changing all the time, but actually I don't think your question's a bad one, because I'll come to why that is in a moment. Let me ask you a question. So if we can say that most stars that are similar to our sun have solar systems associated with them, and it would be that we see, and there's a few billion stars or at least a few 100 million, if not a few billion stars in the Milky Way, then, and let's say 25% of them are like our sun. So let's say a few 100 million, and then at least 50% of those have solar systems. And when there's those solar systems, let's just say another 10% of those could have Earth. You're talking about at least 10 million Earths in the Milky Way, if not more. Now, why is that interesting? Why you're pulling back? Well, I like the idea of looking for Earths, because what I think you mean by Earth: water, carbon, Goldilocks zone, because life on Earth with carbon and this gravity took this trajectory, it might be easier to talk to aliens from an Earth-like planet than aliens from another planet because our metabolism, the time scale on which we're interacting, are going to be similar. It might be that life on some other planets may take a few thousand years even to go through a metabolic second for them. I know that might seem insane, but who sets the time constant? Well, the time constant must be set by the reaction rate and what we how we're able to move stuff around at our pressure and temperature. So I would say looking for Earth-like planets is a good idea if we want to find intelligent life that we can communicate with. But I've just made that up just now in response to your self-criticism because I think it is a valid question. But I just made up the numbers for Earth. But I think I've heard estimates in the Milky Way alone that there are potentially hundreds of thousands, if not millions, of candidate Earths. But that's making some pretty, wide assumptions. Mm-hmm. And they're going to get narrower. The error bars on those will get narrower with observation.
Yeah, but for someone like me who doesn't really know much about astronomy and these things, we're not talking about a handful. We're talking about a much higher order of magnitude.
Yeah. I mean, and there's also lots of things about solar system dynamics, right? Everyone says, "Oh, our solar system is really important because we've got these heavy planets in the outer solar system that cleaned up everything, so Earth wasn't bombarded and all this stuff." And so there's all these kind of everyone's looking at contingent reasons why Earth is special in the center of the universe, and I don't think that necessarily that's a problem, but we need to assess if we are rare. Then we have to cope with the possibility that maybe we are the only intelligent life form in our galaxy, but we just don't know enough.
A couple other topics I'd like to hit in the time remaining. One of them is the origin of complex life. So given that life exists, it took, I don't remember how many billion years to go from single-celled organisms to multicellular organisms. Can we think about that transition using the same framework you're using to think about the origins of life? Because, I would presume that the types of molecular artifacts that a multicellular organism produces are quantifiably more complex than the ones that a bacteria produces, say. Do you think about the transition from unicellular to multicellular life at all? And is there anything you can say about that transition?
Yeah, I'm not as negative on it as you are. I probably shouldn't use the word quite like this because it has negative connotations, but, Earth might just be retarded with respect to its ability to go from single cell to multi-cell. Now, what do I mean? I don't mean retarded as a pejorative. I mean, think about it. Our pressure and our temperature required an ability to take an information to kind of change the way we store information in the cell. And then we took a metabolic change as well, because we put oxygen in the atmosphere. But what about if there'd been oxygen in the atmosphere? What about there was a natural source of oxygen, and so life didn't have to evolve in the absence of oxygen? And so we are there might be quite a wide window on that. So, it may be that on some planets you can get to intelligence really quickly. On other planets, they're just a bit slower, like I say. Retarded, but I mean that from a planetary point of view, and from an n of equals 1, observations, we just can't say anything. All we can comment on is isn't it weird that Earth was quite happy, in one domain until it was tipped into another domain? And I think the hint is this: you need a changing environment to challenge the life form for it to then kick up a level, kick up a level, kick up a level. So Earth was good in that it was stable enough for life to emerge, but it was too stable for life to make the kick up. Life had to actually pollute the environment for things to happen, which I think is kind of interesting. And maybe people are saying, well, actually, you're misevaluating life on Earth. Life on Earth is not, you shouldn't be counting the IQ of life on Earth. You should be counting the ability of life to just form and cope with its environment. And maybe arguably climate change that's coming that everyone's worrying about is going to be another increase in our collective IQ as a species, because we have to out evolve it or out technologically compete it.
Yeah. So I didn't actually think about this before. You're basically saying that you could almost think about a transition in complexity, unicellular to multicellular life, say, as having an activation energy.
Yeah.
To borrow a concept from chemistry, and perhaps on Earth, we actually had a very high activation energy for multicellular life simply because we didn't have oxygen and other things around.
Yeah. Earth was too, let's not use the negative connotation. Let's say Earth was a bit boring, right? And so it took a while for things to happen, but maybe there are other planets that may be smaller, have different conditions. And maybe it's a smaller planet that's less boring that get to intelligence quicker because the planet's going to burn out or something's going to happen. And and you might find a clustering of technological civilizations on Earth-like planets that might be a little bit smaller that have been kicked into that trajectory quicker. But I mean, I've just made this up. It's fun to think about, right? Because I think that we just say, oh, it took this time on Earth, therefore nothing. We don't know anything. All we can say is it's interesting it took this length of time. It could be depressing if we're demanding intelligence to be the pinnacle, but I wouldn't be down on it. I think we just have to be as open-minded as possible in our search.
Mm-hmm. And so I guess this is a good place to talk about the next stage or the next step up, which is, life starts. That's interesting. It's a very interesting transition. Life goes from relatively simple unicellular to multicellular, also a very interesting transition. And then at some point on Earth, you get what we would call minded life that is aware. And I'm not going to use the word pinnacle, but its most complex exemplar is ourselves. And I'm wondering what your general thoughts are on how deep the connection is between thinking about the problem of life's origins and thinking about the origin of consciousness. I know that, for example, I've had Terrence Deacon on the podcast before, and we didn't really get to this specifically, but someone like him would argue that understanding the origin of life is actually very deeply connected to understanding the origin of consciousness, and it's not merely a kind of metaphor. Do you think a lot about that stage of complexity and where consciousness comes from?
Yes and no. I mean, I think that maybe Terence thinks consciousness is more special than it is. I think consciousness is not really that special per se. It is and it isn't. So, but I would agree that I think that the same transitions in information or the same lack of understanding of physics, of the new physics of chemistry and biology that drives the origin of life is also responsible for for consciousness. And I would say origin of life is probably, and a few colleagues, I think Michael Lachman has said this, and Sarah Walker and a few others said, hey, we think that the origin of life is the easiest of the hard problems, with consciousness being one of the hardest ones. So yes, I do think they're similar because there's transitions in information. Because I think that, we really have no understanding about information in the world. Assembly is that first scraping that says, hey, actually, there is information out there that doesn't have to be user dependent, doesn't have to go through you as a god. It assembles itself. When the chain of events can remember what happened, it can make another series of steps. So the information is within that chain. And if you think about it, consciousness as a phenomena probably didn't emerge with a single individual. It occurred in a network, and we then infected each other. Super important to understand that consciousness probably happened that way. And then, and that's also about then how life is literally searching space for correlations, because all consciousness allows us to do is remember the past, be aware of the present, and imagine the future. I mean, imagining the future is one of the biggest hacks that biology's ever made. Because suddenly I can imagine what would happen if I built a bridge that did this direction, or built a skyscraper, or a computer program. These kind of things that then cause material effect in the world liberate me from just evolution. I can do many other things. And I think that from that degree, consciousness is a super important tool for life to be causal in a non-evolutionary, or be a little bit above the layer of evolution. It doesn't beat evolution because it all lives on the layer of evolution, but its effect can be faster.
And so it's really interesting to think about this idea that consciousness is something that we infected each other with. That there was a time when perhaps we had all of the biological hardware, let's just say, that sets us up to have the minds we have today. But it wasn't until some kind of social structure or something emerged that, what we can refer to as consciousness arose, but it arose in parallel in many locations and sort of spread from person to person, almost like an actual virus. Do you think that's do you think that's merely a metaphor? So when people start talking about memetics and ideas
No.
replicating like genes, is there something deeper than a metaphor there?
Yeah, it's much deeper. In fact, it's the mechanism. The thing is, why would think about it? Why would cavewoman suddenly go, oh, I'm now going to think? That doesn't make any sense. Caveman and cavewoman interacting and trying to, oh, there's food over there. There's danger over there. I'm annoyed because I woke up today and I'm wet because it's raining because I live in Scotland, or I'm too hot because I'm living in the, I don't know, whatever, right? Consciousness had to be driven by utility. These things don't just spring out, and utility is communication. So communication, now it doesn't mean that self-awareness is always there, but consciousness, what I mean by abstraction, the ability to then start labeling things and so on. And that was kind of must have collectively emerged. And also, I think it did shape the hardware. There was a minimal hardware where you could start, and then that we selected. So the ability for us to now create memes was entirely evolved. And so I don't think that's loose at all. I think we need to pay attention to it. It's really important. That's why one of the things I keep saying to people about origin of life, why do we care? Well, if you want to understand where humanity is going on Earth, we need to understand the origin of life, because we don't know what life is yet. Until we understand what life is and how the causal chain and the information flowing from that process till now, can we even begin to shape the future? And say, even understand, I would even try to convince people to understand social media today and the evolution of social media. We need to understand the origin of life. And one of the things I'm trying to invent is like a future Twitter or counterfactual Twitter. I'm going to make a Twitter that's basically 2 months older, like that's the universe in 2 months' time. Interrogate that and use it to basically go back in time and seed Twitter now and have this link it may seem insane, but it's not. It's about understanding how at these social levels you get acting down on the biochemistry and it comes up again. So there's a process of causation that goes in all directions, but there is one directionality by time. It's just about layers of abstraction.
Yeah, it's almost in some sense easier for me to think about. So if we come back to this notion of how important selection is, it's almost easier to think about selection at this higher level of the mind, because we have to communicate with each other. And there's a very natural selection process that's determined by whether or not we effectively exchange our verbal communication in order to do something. And obviously we either do it or we don't. Earlier, you touched on something that we didn't go fully into, I don't think, which is the imposition of selection by the physical environment in sediments or the Earth's crust, say, that you're using to do some of your origin of life experiments. Can you talk a little bit more about the relationship between selection, or how selection happens at the level of life's origins, and what that has to do with the physical constraints of the environment?
Yeah, so that is the question. I think when we understand this, we understand consciousness, which is kind of a bizarre statement to make. So really, first of all, I should say that I have some very strange views of time in the universe and the way that determinism works. I'm going to compact a huge amount into about 3 minutes, which will probably need about 3 zillion podcasts if you're interested at some point in the future. But the first point is, I'm a determinist, and the universe had to be deterministic for life to even emerge. Now, that doesn't mean I don't believe in, well, there's no free will, but that doesn't mean I don't, but I do believe there is an unwritten future. Physicists are kind of determinists, and they think the future is written, and it's not. I'll explain what that is in a moment, but I need determinism to work, even at the quantum level, even in the quantum realm, because there needs to be cause and effect. You don't have cause and effect, you don't have selection. So that's the point number one. You have some kind of physical laws or some kind of interactions happen. To happen that, there happens to be 1 arrow of time. So time is not merely an add-on. Time is more fundamental than space. At the beginning of the universe, there was time. That time created space. Now, let's just leave that behind, because you don't need to worry about that. It just helps you understand the causal structure. So you have time creating, and there are events happening in that time, and say boulders bunching into one another. So let's go now to the origin of life and geology. So you have the Big Bang, create stuff crystallizes into matter crystallizes into stars explode, create planets have inorganic stuff on them. That's all kind of determined from the Big Bang, right? Okay, there's obviously undetermined things, but life does something to determination in terms of states available. This is what it really makes us, really is about to fry your brain if it hasn't already. that basically the universe is relatively deterministic but complex. But what life starts to do is to imagine itself in abstraction space. So there is like this greater possibility through selection than the universe could get. So the reason why the universe is not deterministic is because of selection. And this is why I think we have to understand selection goes all the way back to the Big Bang. So what do I mean? If you've got a series of interacting particles, you could estimate what's going to happen in the future on the basis of what's happened in the past. There is no causal structure there. It's just laws. But if you suddenly have selection, where that selection is kind of contained, there's a bubble of that follows around the particle, that bubble, selection bubble, then suddenly things become much more undetermined because there's so many interactions. And you can then see the objects. Let's just now go to the crystal world. I've got a crystal with a milli-Urey. Milli-Urey is just random gunk, but the crystal has an ordered face. So suddenly this random gunk sits on the crystal and doing random interactions, but makes a molecule. Magically, well, not magically, probabilistically, but that molecule happens to be a catalyst for itself. So suddenly a self-replicating molecule gets made on that crystal randomly. And there's more stuff, and it comes in, this molecule goes, hey, I'm quite good at making myself. I'm now making myself. Well, hey, look at you. Before you know it, all the gunk has been turned to this molecule. And in that process, now maybe the molecule makes another accident, goes into another crystal, and it makes another version of itself with another atom on. It goes, hey, I've now made another one of myself, but I've got an extra atom on than I had before. And this carries on and carries on and carries on. Before you know it, you have a molecule with such complexity, high assembly number, can't have formed by chance, but there's a series of events which have created it. And that is what I would call a causal genetic code. And that is the thing that really kickstart life, because you're in this set of adjacent spaces. It's really hard to get your head around. So what am I saying? I've just contradicted my life detector. Said, oh, a molecule in a piece of gunk in a crystal replicating itself in another crystal can have an high assembly number. Therefore, Lee, I've caught you out. That you say that's a lie. That life does that, and I'll say, you see that crystal with that molecule that's made itself in that crystal and a bit of genetic information. That is a minimal life form. That is selection. So what I'm now saying is like, whoa, assembly theory and counting the numbers of molecule parts isn't just about you're alive or not. It identifies selection. So assembly theory is a selection detector. It's not a life detector. It's a selection detector. But the more selection, the more likely you have life. And that was a mind-blowing moment that I kind of realized just a few weeks ago after lots of toil. So I said a lot there, but I think I've kind of addressed your question. And I think it's really important because that selection then gives rise to higher levels. And in the end, abstraction and consciousness. And we now have a history of events. We don't have free will entirely, but we are shaped by our past, which is complex, and our future, what you're going to do next, what I'm going to do next, no one can know because we have all this internal structure and we're reading out. And it's almost like two unwritten books with two read parts. The environment is one set of chapters that are reading you and you're writing it, and you're the other half. And when you get together, you have a series of, you basically know what decisions to make. I don't know if that makes sense. Sorry for the very long explanation.
No, I think this is interesting. I wonder so when we think about selection, I'm going to try and talk about it without using the word in its own definition. But by definition, when there's a selection process, right, you've resolved a set of many possibilities into just one.
Yes.
And that seems closely related to just the basic concept of information encoding, right? It's a resolution of different possibilities into one. Can you maybe connect the dots there for people? And we maybe should have done this earlier, but can you just define information from an information theory perspective for those who don't have that background?
Yeah, I mean, I think we're in this horrible situation with information that we keep misusing it. So there's a perfect triangle, I think, where we have a assembly, entropy, and information. So let's take this all in one. Let's take the easy one, entropy. Entropy just basically tells me the amount of disorder in the system, or conversely, the amount of order. It tells me, the amount of flux and stuff that's going on. Entropy is kind of like almost viewed as evidence of a power source. In fact, entropy is just telling us that time exists and we don't need entropy in the end. But that's heretical, will get me fired and physicists out of jobs and all sorts. But entropy is kind of a fake term that we've used to cope with the fact that we have time and we add time on. But don't worry about that. Entropy is very useful. So stat mechanics. So now information. Information, what is it? So information, put simply, there are a number of definitions, but information is a thing that I can tell you. Okay, so I can give you something I know, which when you encounter that same circumstance, you can act differently. So let's just say I'll say, hey Nick, that you're traveling to, I don't know, a certain town. It's very busy, so don't go with your car. Go on foot. Park your car outside. So you could have done two things. You could have said, well, I don't know that information. I'm just going to go in and get stuck in a traffic jam. Or, I know this. I'm going to park outside and now I can walk freely to wherever I want. That is a piece of information allowed you to act differently. So information is causal, okay? But put a bit more generically, if you take the Shannon electrical engineering and coding, information basically says, hey, you're playing poker with me. I've just dealt you 4 aces or 3 aces. The chances that your 4 players around or the other players around you have an ace, because you have 3 aces, that you have reduced your uncertainty in that. So information is really at its core about reduction of uncertainty. So that's a very important thing to have. But the problem with information in our universe, without living things, there's no information. That's really kind of messed up. What I mean by that? Well, if there's no living stuff, there's no encoding, there's no decoding. So I can't tell you something. So that's why the assembly is lower, is more primal, more fundamental than information, because assembly is in the causal chain. And then assembly allows you to build life and build objects, and then we can communicate. And then this phenomenon, information, kicks in. What the problem we have right now is physicists are using information theory built for communicators to look at origin of life and systems, and it's not fit for purpose because it's meant for something else. So you have to put yourself in the causal chain, then you get assembly theory. When you're above the causal chain, you are information theory.
Well, this is definitely the first podcast conversation where I want to listen to this one twice after we're done, once in a sober state of mind and once not. I'm wondering if you have any non-technical artifacts, like a book or anything that you've written that people might go to if they're unfamiliar with your work so far.
Non-technical artifacts? No, I'm not very poor at that. I suppose there are a whole variety of other podcasts out there. There is also some kind of videos. There's Inorganica, which was made many years ago, which is fairly embarrassing, but fairly descriptive about what one's trying to do in terms of looking for life. And there's also, I suppose, a quite a long set of conversations I've been having in the public domain with people on Twitter, on blogs, increasingly on Clubhouse about these concepts. I am starting to write more generally. Now I understand how to put it in such a way that sorry, you can cut that. It's my son. I should have texted them to say, hang on one second, right, come back again. So I'm doing increasingly lots of discussions on blogs, on Clubhouse. I'm starting to get to the point now I understand what I'm talking about in a more general sense. So I'm writing some more general texts. I might even write a book or two one day. But right now, I'm just trying to get this triangle of ideas, like I said, in entropy, information, assembly, right? And then understand all the way back that life is not a one-off. Life is driven by selection. Selection is a fundamental objective process that occurs in the universe. Selection is the way to get to information and abstraction. but it has to go through this process that we don't yet understand. And that is really fascinating because we're just at the beginning, right? I don't have all the answers. In fact, I don't have any. I just have questions, but a more non-biased look. And the way that I'm developing my theory, as I said at the beginning, is through experiment. The assembly theory, I just didn't dream up one day. I thought of the mass spec experiment and thought, oh gosh, I can tell complex molecules, Taxol, which is a complex drug made by a tree bark, from a random molecule that I might find by accident dead on Mars. And when I realized those two things, and I kept obsessing about it and said, well, maybe it could naturally occur, but maybe I could naturally, flick a coin and get 20 heads in a row. How long would it take for me to do that? And you could calculate it. Say, oh, that's not probable. Now let me say I can flick 20 heads in a row on demand. Everyone would know I'm cheating. And when I realized that my colleagues, when they said, oh, maybe you could get 20 heads in a row, they realized actually, when they play that to themselves, that doesn't work. So I know the theory's right. And I think that what I've tried to do is to try to help NASA, because the problem that we have going back to the origin of life is we have a series of arguments based on ego and narrative, a series of definitions based on ego and narrative, and a series of what is the most important way to look at it based on ego and narrative, right? I mean, of course, science is based on ego and narrative, but what I wanted to do is unify people. So I decided to do one thing, is to take a leaf out of high energy physics. We have the standard model. That's the theory for matter. Works brilliantly. Now, with the standard model, we can calculate where we might find particles. So we do that. Now we can build a particle accelerator. And you know what we went and did? We said, okay, given the standard model, with our calculation and our experiment, we expect to see a peak here, here, and here. We went and did it and found it. We even found the pigs. So here's my idea. I have an idea that the origin of life, or life in general, requires assembly. I can calculate roughly how much assembly we need. So now I'm going to go make a robot. And I know roughly how long to look before I start seeing selection. So I build my robot starting random, and it starts random and can feed itself. So it should get non-random over time. And at some point, it should cross the threshold where I can detect assembly. And that will be the point at which we invent life in the lab, objectively, demonstratively. And that is so exciting because it will happen.
Lee Cronin, I think that's a great place to leave it. Thank you for joining me today.
Thanks very much, Nick. It was a pleasure.